So I'm going to talk about this topic, small RNA and epigenetics. So I'm going to give you some insight about the mechanisms and why they are important. Why are these molecules important? So in the lab, we use all these model systems. So these are all fantastic model systems. And my lab is in NCBS. So you can reach me in this email. So in case you are interested in working with us or you want some information. So we continue to depend on plants. This is not a big surprise. Everyday life from morning to evening, we end up, depending on plants for food, for clothing, for medicines or shelter. So plants have been providing all these since our own evolution. And now we have even like a urgent need. We want climate resilient crops. And the only aim is to get higher yield in all seasons, in all conditions, that too in a sustainable manner. So this is like asking a two-wheeler to carry 2 tons of weight. So now, just like in everything, we need to know why and how. Why plants make all these things and how they manage to make these things. So that's what I'm going to discuss in the first part of this talk. So now we need to increase this yield, and it is very clear that incremental knowledge, just changing the seed size little by little, is not going to be very helpful. So we need some out of box approach. Now, why plants make all these things? Plants make all these things because they want to interact, collaborate with animals. So if you go back to the Jurassic times when we had all kinds of nice animals, we didn't add flowering plants. So flowering plants came in the last 100 million years ago. So they are like abominable mystery even for somebody like Darwin who had answered for everything. They came in no time, but they conquered the world, literally changing the landscape. And now in every campus like yours or mine, you will see predominantly flowering plants. So this last minute change is because they started interacting, collaborating with the animals. So they tricked animals to spread them and their genes by bribing with colorful flowers and nutritious fruits. So the seed spread, there was modifications in the structures to allow such a thing to happen. And animals also benefited. They really changed. There are newer forms, including us in this time. So this is like a real change in the history of our Earth's history. So now why they make all this or how they manage to make all this. So not all plants really behave the same way. Some plants really were very clever. They targeted us. We could move easily, so we could move the seeds. We are also capable of doing all kinds of nasty things. We removed all other things, all other plants, all other animals just to pamper few plants. And this is the birth of modern agriculture roughly 10,000 years ago. And here I have put the plant pictures of all these cereals. So cereals became the key plants because they produced carbohydrates that doesn't go bad very quickly, unlike protein. So one could store these seeds, and this was the birth of so-called agriculture, fencing, birth of countries, so on. So this agriculture literally even then changed it became like a watershed moment in our history because we wiped everything else to make space for these plants. In this bargain, there is more grains for us, so we also proliferated more. Our population increased dramatically, but the dominance in numbers and pampering, they go for the crops. There are crops such as cauliflower. You can't simply grow in the wild and expect a fruit. They need pampering from us. So if we go back and see what has changed in the plant, so one, why we sort of know how this change has happened. So if you look at plants such as maize, it's a product of artificial selection by us. So you can see in this picture how there are two types of wild species of maize and the current maize, which is almost 1.5 foot long. It can really cater to almost, it is a meal by itself. So this is because we could make all kinds of hybrid. We ourselves are hybrids produced, resulting by combination of other primate species. So if you go back to the comparison, so between maize evolution and that of transport, so I mentioned that animals also evolved a lot. Animals put their money in two things: one, locomotion, and second, on consciousness. So we are all very fond of this locomotion. See here what has changed in the evolution in the transport in the upper panel and evolution of maize. Both are dramatic, both really took off in the last few decades. So why the evolution of, for example, transport evolved in the last few decades is because of real change in science and technology. The same is true for maize, and that's when we really started doubling, tripling, quadrupling yield of these crops. So one major change in the last few decades has been biotechnology. This is transforming agriculture. It is part of the domestication process. So it has two meanings. It refers to innovation in use of plants to make products of use to us. Second, it is use of genetically modified organisms. So with these technologies, last few decades has been really surprisingly, very good for us, because we ended up producing a lot more food than we did ever did that much in our history. So you're all familiar with this tissue culture. So banana, literally every banana that is used elsewhere, not in Kerala, because Kerala you don't do that much of tissue culture for banana. So everything, every Cavendish, every one of those bananas, they are all result of tissue culture. So while this is good for rapid multiplication, it doesn't bring anything new to the table. So it's good for dissemination of same type of material. But what has changed is, for example, something like grafting, it can break the species barrier, and this can combine properties of two plants and two stages of life cycle. For example, you can combine tomato and potato to make this nice plant called pomato, see how it looks, or combination of different characters of fruits, or early flowering, etc. But the problem here is, you can only do very closely related species you can bring together. And second, it is good for only fruit crops. Okay, so the demand for fruit crop is not much. It's not like we can only consume them. But again, such a grafting did help us to produce much more food. So, but the key here in the last many decades has been the hybrid vigor, the magic that feeds us. We ended up making literally everything that is in our plate, that's in our what we consume, has been a result of some hybrid. So if you look at orange, the now we associate orange with the color, but orange itself, the wild orange, real orange, is green. It's not orange. So just because some chlorophyll genes are now messed up and they make this orange color, now not just fruit is colored orange, everything of the similar color is named as orange. So the such a hybrid vigor is what we see, what has made us to again produce a lot of food. And but to make this breeding, it is very slow. It uses brute force. For those of you who don't know what is brute force, if you forget your 4-digit password in the mobile, if you try each combination, how long it will take. So basically that's brute force, and that's what breeders do to get these new characters. And more problematic is results can be undesirable. Let's say if you're trying to get more sweet fruit, you might end up getting bitter fruits. So to bypass all these problems, we have the genetic modification. So it has the potential of increasing the quality and quantity of food worldwide. So papaya industry came back from brink in Hawaii just because one person's efforts. Dennis Gonzalez from Cornell, he put a one gene of the virus into the plant, and now plants are suddenly tolerant, resistant. Same is true for golden rice that you all have heard. But the difficulty with genetic modification is it attracted, unfortunately, poor public opinion. It needs expertise, it needs basic biology information. That's also lacking for most of the crops that we use. So what is why I said it's unfortunate that there is a public opinion, is because most of the people who work in similar topics, we know that agrobacterium, which does this magic thing, is a common soil bacterium. So you must have seen all these mango trees where you can see these tubers. These are all result of agrobacterium's tumor induction. Agrobacterium has transformed its gene to this plant, and you have these tubers. Did you ever thought you are probably eating a transgenic mango? No, right? So in this scenario, so the bacteria takes out one of its plasmid, and in the plasmid there are genes to make these tumors, which will be auxin and auxin biosynthesis genes as well as cytokine biosynthesis genes. And such genes can be, through this plasmid, can be introduced into plants. Bingo, there you have the transgenic plant. Now, such a thing as is not artificial, not created by man, it is already there, because look at sweet potato, it is a naturally evolved transgenic plant. Uh, 8,000 years ago, sweet potato plants did not manage to make a tuber. They made a tuber because agrobacterium, which is in the soil, moved, introduced its gene as transgenes to the plant, and now we have the tubers. Okay, so this was published many years ago in PNAS. So whatever it is, the result of this is abundance of food never seen in history. So we still have unequal distribution. For example, some part of the world lacks food, but just 2 million US farmers can generate food for 2 billion people. So we are only around 8, 9 billion people. So, but what happens to that excess food? It is converted into alcohol or biofuel or many other, or food for other animals, domestic animals. So, but because we can make so much food, we can also choose consumption of high energy food regularly. This has both good and bad effects. So now, if you see in this graph, production of rice is really, really dramatic in the last 50 years or so. And what is the mechanism behind rice domestication? How rice changed to make so much of food, while its area of cultivation actually did not increase but reduced in the last 50 years. So many groups are very interested in understanding what has happened, what has changed during domestication. So the wild species of rice is called Oryza nivara, the one which we eat. Rufipogan is another species which is the precursor of both Indica and Japonica rice, but for Indica rice it's mostly Oryza nivara. These plants are still found in the edge of lakes, sort of like pythons. They are prostrate, they'll be sleeping all the time, they make few leaves and few seeds, and they are semi-aquatic. Okay, so how these plants got converted into really plants that can produce hell lot of seeds, sometimes up to 250 grams of seeds per plant. How did they manage to change? How we manage to change? How they change themselves? So uh, people have tried to study, and obviously everybody will think maybe there is a change in the genome, in the DNA, because genome is where it's like an archive for everything, all kinds of information, including social, historical, and cultural information. But what we found is that if you compare wild species of rice or resinivora to what we eat, let's say a Kerala variety, there are very few genetic changes, very few changes in the DNA. So just to give you an example, so we know around 200 yield related QTLs mapped so for rice yield, related to yield. So QTLs are large unknown regions in the DNA, but we know only 8 genes. We don't know literally what even changed, what made the change in the yield. So what we also know is that all these change in the phenotypes, you have bigger grain, smaller grain, color grain, flavor or not, these are all not related to genetic changes. So we thought maybe there is role for small RNA and epigenetics. Now, what is small RNA and what is epigenetics? So if you look at DNA, which is like a book of recipes, so each copied RNA is like a copied recipe from book of recipe. So which can be, you can use it, you can throw it, you can degrade it, and from this you can make a dish which is called protein. So this, from making from book of recipe to making dish, is not straightforward because there are lot of changes. It's like whether the cook can pick the right book or the right recipe or make in the right way. So all these checkpoints exist. That's why if you simply have the DNA, you can't have the right protein. So major checkpoint for this is small RNA, because this is a bit of this RNA, and it can go and tell which RNA should be making recipe, or whether RNA should exist or it should be destroyed. So a small work connected to this got Nobel Prize in no time, in like 4 years. Usually people end up waiting for Nobel Prize for long, long, long time. So what is epigenetics? Epigenetics is nothing but changes to the DNA, which is some kind of a modification in the methylation in cytosines, or DNA, as you all know, it is wrapped around these histone bits. So these are all small proteins where DNA is tightly bound, and bits of this protein, they hang out. And wherever it is hanging out, they can undergo translation, post-translational modifications to the protein. The way the protein functions will be different. Because of these two changes, now the DNA can be really compressed or really relaxed. And this is a big change. This is like a physical force. If the DNA is relaxed, the it is easy to copy the RNA and make the protein. If the DNA is really compressed, then because of these modifications, epigenetic modification, then it's not easy to make the recipe or read the recipe from this book of recipes. So you have to remember two proteins that are required for making small RNA. So one is a dicer, which picks this long double-strand RNA or RNA substrate and cuts into uniform size bits of different size, 21, 22, 24, and so on. And these kind of machines, these are cutting machines, are more in number in plants than in other organisms. The second protein is called Argonaut. Here the protein, the bits of RNA made by dicer is picked by this protein, which is in yellow. And once it picks, it can look for complementarity in Watson-Crick complementarity. G should bind to C, A should bind to T or U. So if it finds this complementarity in that just 21 nucleotide long small RNA, it's enough to go and regulate the target. What does the mean regulation means? It means everything. It can make them help to make the protein, or it can destroy, or it can shift it to somewhere else, all kinds of changes. So these proteins do this magic are called Argonaut proteins. So they are named nicely. Somebody who looked at the mutant of one of the Argonaut in Arabidopsis thought the plant, which is shown in the middle with this thin, long, elongated leaf, looks like something called Argonaut argo, an octopus, and they named it as Argonaut protein. So together they make up the small RNA pathway. So this is a pathway which makes small RNA and helps it to act on the cognate DNA and RNA. So here RNA gets converted into double-strand RNA substrate. Substrate double-strand RNA is not commonly seen in systems in any eukaryote. It is chopped by dicers to make these small RNAs. These small RNA get into Argonaut protein, and once they get into Argonaut protein, they can go and cut the RNA into two bits. So this Argonaut does what is called a splicing. It doesn't cut both strands, it just cuts one target strand in a very precise place between position 10 and 11 in the complementary region. So this is how most of the plant small RNAs, especially microRNAs, which are one category of small RNAs, work. But what is interesting is certain size small RNAs with certain Argonauts can go and target DNA, especially in Saccharomyces pombe and plants, but not in other organisms. And this modification can induce DNA methylation and histone modifications that I explained 2 slides ago. This modification now can be termed as transcriptional silencing. Once this change takes place, now the DNA is packed so much it is not amenable for transcription anymore. What is even more striking, once you induce such a change in plants but not in other organisms, such a change is heralded in the subsequent generation, almost like what Lamarck mentioned in his landmark study. But unfortunately, he took the wrong example of giraffe. He should have picked plants as a model system. So the result is, if a plant is undergoing something, it can be a change in temperature, that information through small RNAs can be introduced as epigenetic change. And now the epigenetic change can be passed to the next generation. So the next generation, the plants have this transgenerational memory. They can cope with the stress and so on. That's why you really don't see a lot of plants really not coping with all these changes that we have thrown at them. Okay, so they have the ability to innate ability to change to the situation, counter the stress, and live forever. So is this well documented? Do we see this? We see this every single day. Only thing is, many of the mechanisms we don't know. So you're all may be familiar or maybe knowingly or unknowingly, colorless never ripe is a gene. It's a mutant, which is we call it as epimutant. I'll tell you what this change is. And this is the one which makes tomato not to go for ripening. In the bottom scheme, you can see if this gene, which is shown in gray box, if it is expressing, the protein is made, you get the red tomato fruit. But in the other upper part where gene is not expressed, it will the fruit will never get ripened for months together. What is changed here? Is there any change in the DNA? No. What is changed is around 2 kb upstream of the ATG, there are some cytosines where the DNA is methylated in this colorless never ripe, and that change 2 kb upstream, just around 11 cytosine methylation is enough to stop the transcription of this gene. The recipe is not read, gene is not made, converted into message is not converted into protein. So when protein is not made, the tomato never ripes. Such examples are hundreds of such examples are known. So there are in the literature, and I think we should be all aware of this. So this is a epigenetic change, no change in the DNA, but there is a huge change in the phenotype and the way plants cope with stresses and other cues. This is also seen commonly in animals. So there is a, for example, those of you are familiar with ants, you have different type of ants, minor worker, major worker, and so on. All of them, they also have this epigenetic change. The DNA is same, but it is read differently. You end up making different protein because there is histone modification. What about others? All the honeybees. You are really maybe even more familiar with them. You must be knowing royal jelly, which is secreted by the queen bee, so it can make the next queen. What does it do? It produce histone D8 acetylase inhibitors. They change the histone modifications. Now you can have the same DNA either making protein or not making protein, and it is dynamic. It can suddenly select a new nurse worker bee and convert into a queen just by doing this. So in plants, for example, the hybrid which I mentioned, so we almost a decade ago we found that hybrid changes, when there is a change in the hybrid, if the hybrid change is heritable in the subsequent generation, that's also because of DNA and epigenetics. So follow these pictures in the tomato. Solanum pennelli is the wild species of tomato. You all know M82 is a cultivated tomato. F1 has shape, size, and color exactly in between both the parents. But if you backcross F1 to M82 for 6th generation, so you're talking about great, great, great granddaughter lines, you suddenly see new characters that are not present in both the parents. And for example, beta carotene is high in this one of these great, great granddaughter line, which is not present in either the wild species or the cultivated species. So these are new characters, and such new characters, which are the ones which made our hybridization, hybrid vigor, and heritability, they are all because of DNA methylation and histone modification. So with all this information in mind, we thought if the DNA change is minimal in rice, could it be possible that there is small RNA or epigenetic variation? To study this, we took two different species of rice. We collected hundreds of land races, including from Kerala, and many cultivated rice lines, all from official sources. We did phenotyping for 125 characters, then selected 12 lines, including two wild species of rice, and started seeing, is that changing the genome? Is that changing in small RNA expression, both small RNA and long RNA? Is that changing DNA methylation? And is that change in histone modification? When we did this, we got hell lot of candidates where these such changes happen. One such example is analyzed by Shweta Chenna, who has just finished her PhD. So what she shows is that wild species of rice called Oryza nivara and to some extent Oryza rubipogan, they have a unusual abundance of 22 nucleated small RNA that are otherwise not seen in plants at all. So if you see in most plants, you don't see except in that blue bar, which is Oryza nivara, suddenly get a lot of small RNA. In the bottom graph, you can see lot more 22 nucleated small RNAs which are circled in wild species, to some extent also in Oryza rubipogon, but not in land races and cultivated lines. So where are these odd category of small RNAs coming from, which have not been documented from any other plant? So when we looked into the 12 chromosomes of rice, so we found chromosome 2 has a candidate region which makes 140,000 reads per million. That's basically plenty of small RNA in wild species, but not in cultivated species. We mapped between certain markers. We found this new microRNA. This is an authentic microRNA. It went to the right Argonaut. It went and targeted gene. Now, what does it target? So if I mention that in wild species it is very high, first we have to confirm that it is indeed true. So when we did a northern analysis looking for this specific microRNA, you can see very high signal for Nivara, which is the wild species of rice, intermediate levels in cultivated in the land races. I think a couple of them are from Kerala, Chomala. I'm sure you have heard about this. Then almost no expression in the cultivated species. Correspondingly, wild species are semi-aquatic, they need stick to stand, they hardly make any grains, whereas cultivated species, they make a lot of grains, they also can withstand even mild wind. Now, what are the targets of this microRNA? I mentioned that microRNAs go and cut the target into two, so the target RNA cannot make the protein. So the targets are genes called laccases. So they come in a pathway called phenylpropanide pathway. Here, this phenylpropanide pathway starts from phenylalanine. Humans use this pathway to make dopamine for our memory, but plants make them to use flavonols, anthocyanins, very important compounds, lignin, which is the second most abundant polymer after cellulase in Earth. And this, to convert lignin, you need the from the alcohol, whether it's synapyl or others, you have to convert these monomers into polymers. That job is done by laccases. So laccases are present in every plant. They have to be present because then only plants are going to come and they can grow stronger and reach the light source. So lignin provides mechanical strength. So this is the kind of gene which the microRNA targeted. There are 24 such laccases in rice, almost all of them uncharacterized. We found that this microRNA can target half of them, cut their RNA into pieces, and in these laccases are not produced. So what we see is that I mentioned that wild species of rice, you have high expression of microRNA. Can somebody tell me what will be the expression of laccases in wild species? Will it be high or low? Just to check to see whether you are listening to me or not. Anybody has say you can try? So in wild species, microRNA is very high, right? In cultivated species, microRNA is low. What do you expect? In wild species, the target RNA should be highly expressed or not? Nobody? Okay, do you hear me or you have lost me?
Yes, sir, we can hear you.
Okay. So now in wild species where microRNA is high, okay, do you expect its target to be high or low? Okay, are you all hungry or angry for some reason? So the microRNA is very highly expressed means it's going to get into Argonaut and cut the target RNA into bits. The target RNA expression should be low. Do you follow me or no?
Yes, sir.
Okay, so in B, you can see the expression of laccases very low. All these dots represent expression of the gene in RNA-seq experiments. very low in wild species, intermediate in land races, and very high in cultivated species. If the protein is high, do you expect more lignification or less lignification? I hope among the 70 plus people, somebody is able to answer this. So if the protein is high, do you expect more lignin or less lignin? So what did I say here? Lacase helps to make lignin. So if you have more lacase, less lignin or more lignin? Okay. More means more. That's what you see in A. When you quantified lignin, more lignin in cultivated rice compared to wild rice. Wild rice doesn't make much lignin. So in sections of stem, we again see all the coloration. The pinkish red color comes from fluoroglycinol staining. Less lignin in wild species, very high in cultivated species. So these are all glycoproteins, so we could pull out using Concanolin columns. Again, you can see in E and F more protein in cultivated species compared to wild species. So N is Nivara there. So all suggesting that in wild species, which are I mentioned, they are all semi-aquatic, they don't need much mechanical strength. The microRNA expression is high, lacase expression is low, so lignin is low. So we thought maybe this must be a real key rice domestication event, because in wild species which are semi-aquatic, you have a lot of microRNA, lacase expression is low, and low lignin means prostrate plants, closed panicles, and seed shattering. Seed just falls off. But during domestication, we converted them to plants which are having high expression of lacases. That means they can support panicles, grain filling, and shatterproofing. So that's what you see in literally all the high yielding cultivated rices. Here microRNA expression is low. So we know why the microRNA expression is low, but I'm not going to go through it today. So during domestication from wild species to cultivated species, now the microRNA levels reduce, lacase and lignification increase, so we have more yield. Now we have to still prove this. How to prove this? We took this high expressing microRNA, put into the cultivated background as a transgene, and then try to see what happens to the plant. So when we did that, we overexpressed in two different cultivars of indica rice and found that plants now are falling off. They don't have the mechanical strength, and they made a total of 0 seeds. Okay, so if you express microRNA, it's already enough to make the plant look like wild species. But if you overexpress the lacase, some of them, the plant is very happy. In the bottom panel, you can see, and it also made much more lignin, and that means it made much more grains in the seed setting rate dramatically improved if you express lacases. So this work got published, and then we thought, how come nobody has found this key regulator? So I mentioned there are 200 such QTLs in rice for yield from many decades of work all over the world. We went back to those 200 QTLs and started overlapping with lacases and microRNA. We suddenly got 26 of the remaining QTLs, which are not known, mapping to lacase and microRNA. So basically, till then we knew only identity of 8 genes. In one go, we could find all these so many genes related to yield in rice. So the same is true for other domestication events, such as in selection of colors in fruits. So fruits, even flowers, is a promise of food. That's why men also are interested in flowers. So in fact, whole country of Holland, or the Dutch people, they were really crazy about tulips. Even today they are crazy. So the first one is this tulip called Semper Augustus, which is a very unique anthocyanin pigmentation. Or any fruit which is colored, if you all given a choice, you will go and pick the most colorful fruit for your dessert. So not just humans, even animals such as birds, they also like to choose fruits which are more colorful. So this is the bargain which plants have made. They make this compound to help Either if it is for in the flower, it is to attract pollinators, or if it is in the fruit, it is to attract animals for fruit dispersal. So we found out that some of the grape varieties have very high levels of certain microRNAs. So to do this comparison, we took three different grape varieties. Two of them, top two, have very high levels of anthocyanin, which are flavonoids with the color, and Dilkush, which is the green grape, it doesn't make anthocyanin, but it makes a lot of flavonols. Okay, so flavonols plus anthocyanin together are flavonoids. Okay, so we from this we did whole genome sequencing of small RNA, RNA-seq, and so on, and we finally found a gene called MIB114, target of the microRNA. So in phenylalanine, the same fun pathway, phenylpropanate pathway that I mentioned, it makes also flavonols and anthocyanins. So MIB114 sits in that branch point. If it is expressed, dihydroflavonols will get converted into flavonols, as in green grapes. If it is not expressed because of microRNAs, you get anthocyanin. So basically, to study this, we had to do transcriptome, we had to do metabolome, we had to do proteome, and found this gene called MIB114, which is shown in the red arrow. And if it is expressed in models like tobacco, it suddenly makes a lot of flavonols in the one of the bar diagrams you can see. Whereas other genes which are like MIB-A7, if you express them, they make plenty of anthocyanin. If you express MIB114 on top of A7, which makes a lot of anthocyanin, you start getting more of flavonols. So that's why we ended up saying if MIB114 is repressor, which is a repressor class, new class of repressor which we identified, if they are made, fruits are going to be green. But if they are not made, if they are cut by the microRNAs, now the MIB activators can make colorful fruits. So this seems like another example of coevolution because we all selected for colorful fruits. And this is not just true for grapes, but molecular signatures show that apple, strawberry, at least in peach, you can see the same signatures. So once this paper came out, we got at least 10 papers showing coloration is linked to such MIB repressors and microRNAs in plants such as pear, potato, and so on. Okay, uh, so it's not just such microRNAs alone are needed for domestication changes. Even expression of genes involved in silencing are good enough. So there are a couple of Argonauts which nobody has studied in monochores. So in dichores, there are only 10 Argonauts, but in monochores, you can have 20 plus Argonauts. We looked at two of them. Both are actually differently expressed between wild species and cultivated species. If you overexpress them, some of them you suddenly got a lot of yield, such as in Agos 17, or if you remove them, you started getting male sterility, suggesting that every yield related phenotype could be because of domestication associated small RNA or epigenetic change. So where we are now, so we are in the, so we have to feed the global population. So this is just like your phone, you have 1G, 2G, 3G, 4G, etc. in the evolution of crops. So until now, so 1900 is when we had the green revolution, mostly because of changes to dwarf plant type, selection of plants. Then we got the biotechnology, which I explained briefly until 1992 till now. So there is marker assisted selection, genome prediction, etc. So this allowed us to produce much more cereals, and of course this helped us to increase the world population, or cater to the increased world population. What we are in this, we are now in the 4G, so we are in the design breeding stage. Here we can edit the genome, so the DNA is there. You can precisely change one or few bases either to express a protein or remove a protein, or design by big data and knowledge driven breeding. So that's the stage we are in. We are already making a lot of crops, and but the demand is way high because you can see the projected world population. So what can we do? So the most innovative plants of 2020. So we are yet to get the 2021 list. If you look at them, these are examples of 20 plus plants which are well known. Most of them, so this aliote potato is almost like a sweet potato, except that it is not sweet. So it combines the properties of potato and sweet potato. Sweet potato has vitamin B12. So you can see you can improve the nutrition aspect of these crops. Maybe the size, such as in this maize, where the person is standing to this tall maize variety called A170 corn, but mostly improved nutrition, right? Not necessarily anything to do with the climate change. So that set of plants are yet to come. So this is a bad news for everyone. But the good news is, so what we are doing now, so what is the roadmap to make this generation? If we take 5-10 key genes, we can make a quickly a domesticated crop from any given wild species of crop. I'll tell you why this is important in 1 minute. Now, if you make specific pathways modified with few genes, such as the one we have done for flavonol engineering or lacase engineering, you can try alter a trait which can take 5 to 8 years, even for difficult crops such as rice. But the demand is this: ideal crops should have multiple such things combined into same plant in many layers, including, and they should not be dragged down because of changing climate. So we can do this quick de novo domestication even quickly now, because we can make what is called as de novo domestication. This is shown by Chinese researchers recently. By changing 4 of these genes, you can suddenly make a wild species a cultivated species in 2 years. Okay, but what about the climate change? Lot of labs, including ours, are looking at climate change. So this is one such example. So we have a plant now in rice. It's a genome edited line with CRISPR-Cas, probably the first of its kind in the country. So these plants, if you see, they have thinner leaves, they are resistant to stress, they make much more secondary metabolites, they also make more yield. And these plants don't change because they are thin and they don't have open to allow dehydration of plants, so they are much more tolerant. I can't give you the name of the gene yet. So over the last many years, we are working on these pathways. So we have identified several new mechanisms, also employed many of them to improve crops, including improving the climate resilient plants. So this is only one aspect of it. So that's only that needs some expertise, some specific well-trained people, which can be some of you if you wish to. But what everybody can do is this. So domestication of a new crops that are climate resistant, if that is the goal, we can't simply look at the 300 plants that we eat now. There are at least 3 lakh edible plants. Many of them can be converted very easily to make them palatable to us. We may have to have also a social change, cultural change. So that's something which is with us. This is really needed because uh, uh, so what is likely to happen now is already happening. So we have huge reduction in the crop. Most of the pulses are bought from Canada or Australia. So by 2050, we will import more than twice the amount of food grain than we are now. And we are already spending a lot of money. We don't have any empty area in the country. We are somewhat used up almost all the area. We are actually removing the remaining forests for agriculture. What can be done? Crop diversification. This everybody can do. We have to start eating things which are actually quite tasty, but nobody else is jumping. You can be the change. So we have to efficiently use the water use, which we are trying but haven't succeeded well. Improve the soil management practices. Again, we are trying, we are not successful. And development of climate resilient crops that can be done. So we are doing, many people are also involved. Let's see where we go. But I wanted to show these slides with this. So many of this need more knowledge. For example, apple is only 10 times 10% of nutrient compared to guava, and I'm sure you won't pick up a guava if you are given a choice. So apple doesn't keep the doctor away. This is a lie started by some of the US people 100 years ago when the Prohibition Act came, and they could not make alcoholic drinks out of apple. So they said apple a day keeps doctor away. So it's a lie. So actually, most of our fruit crops are much more nutritious than what you see in the superhead shelf, in the supermarket shelves. Also, something like TEF and many pulses. So TEF is a favorite of marathon runners. It's in Ethiopia and Kenya. That's why we have some, marathon runners who can run really long. This is the one which you are now importing to feed to our marathon runners. Similar, our ragi is not very different from TEF. So these are the changes which we could do to get nutritive food, and all these crops are also naturally climate resilient. With this, I would like to thank people in my lab. They call themselves Epipepis. Never understood why. So these are all of them do fantastic work. Their projects, I hope I have a chance to explain. So my source of inspiration is David, who is the discoverer of small RNA, and Professor K. Velthambi, my PhD boss. Fantastic collaborators, lot of facilities, and a very nice place in SBS. So I'm supported by funds coming from DAE through TIFR, that's our mother institute, parent institute, and from DBT and EST. So I would like to thank Professor Supriya for inviting me and giving me this opportunity to talk to all of you. I see the number from 75 has reduced to 50, but I hope those of you who are listening to me, you have learned at least something. Thank you very much.